Hello, everyone. Today I'm going to give an introduction to SenseAI, or SenseAI for short. SenseAI is a spin-out company from the University of Liverpool, where it is focusing on the use of compressive sensing for all types of imaging applications. One of the areas that we're focusing on at the moment is in electron microscopy, where by acquiring a smaller number of pixels in our image and using a form of compressed sensing called in-painting, we can recover the full image for much lower dose and much quicker. So just to give an example of how the approach works, let's start off by talking about sparse sampling. So sparse sampling is pretty much the way that it sounds. Typically, when we're doing a scanned image, we do a raster scan, which is much like a word processor works. We type all the way across to the right-hand side, and then we flip back to the beginning, one line below. Now, if we want to randomize this scan, we can do what we've got in the middle, and so we can have a random scan instead of a raster scan. If we want to subsample that random scan, you can see on the third in C, we have an example of a random scan. And then because we have hysteresis in electron microscope, we have to do an implementation of the random scan, which we call line hop. And that line hop basically allows us to do a random walk and to get a randomized subsample scan of the image. So our goal is to apply or acquire as few pixels as possible, and that speeds up the image. But to make it practical, we also have to be able to get our reconstruction of the full image as quickly as possible. And what we're aiming for is to have a video rate reconstruction so that when you acquire a sparse sampled image and you do compressive sensing and in-painting, you get an image in the same time as you would normally get a full scan. So let's talk a little bit about the reconstruction. So here's an example of something you might see in a basics physics class or electrical engineering or signal processing class. We have a complex waveform on the right, and that complex waveform is made up of the dictionary elements that we have on the left. And these are just sines and cosines. And in the terminology of compressive sensing or dictionary learning for compressive sensing, those sines and cosines are the dictionary elements. Now, if we know what the dictionary elements are, we can actually get rid of a lot of the signal in our complex image or our complex structure and effectively still reconstruct the image very accurately. And we can get down, it's only when we get to about a few % of the signal that we start to have small differences in our reconstruction. So what this is telling us is that effectively, if we know what the dictionary elements are, we can reduce the amount of signal we need to acquire, and we can get an accurate reconstruction. Now, in this case, there's no compressive sensing or artificial intelligence involved in the process. We know what the dictionary elements are, and so we can go ahead and reconstruct the signal. In the images that I'm going to show from the electron microscope, the AI comes in because we don't know what the dictionary elements are up front, and we don't know what the complex signal is, and so we have to solve together to get those to work. And we use a process or an algorithm called BPFA, or beta process factor analysis, and that allows us to get this process moved forward for solving an image when you don't know the dictionary up front. So what we have to do is to figure out what the dictionary is. Now, the process that we use is to start from something like a wavelet or a standard dictionary elements that allows us to reconstruct the image, and then to modify that based on the structure in the image, and that's what you get with the BPFA output. So effectively, what we've done with SenseAI is to focus on how do we apply the dictionary to allow us to get our quick reconstructions. And this is the key to doing the compressive sensing. We have to be able to do it quickly and efficiently through either learning the dictionary or transferring the dictionary from another part to be able to interpret the image. So here's an example of a reconstruction of a live image. This is an aberration-corrected stem image that's obtained from Harwell in the Rosalind Franklin Institute, and it's just silicon dumbbells. And you can see that we can get a very rapid reconstruction. So this is a live reconstruction of a 25% subsampled image. Now, because we're learning the dictionary from each individual image, we're getting to the point where we don't have to do learning like you normally do with artificial intelligence. That means we can change the focus, we can change the magnification, and we can still reconstruct the image very accurately. Because we're not doing a machine learning or a deep learning approach to reconstructing the image, it also means we're not subject to hallucinations. If we can't get the information across, you'll just see a blurry space where there's not enough information to get the reconstruction. The other part of this, of course, is that once we've reconstructed our full image, that means you can do all of the other methods for image processing at the same time after you've got your full image. Effectively, the approach that we've developed with SenseAI is to reconstruct from a subsampled image into a full image. After that, you can do whatever you want to the image. So it also works for doing 4D STEM experiments. And so here we have an example of a 4D STEM experiment. You can see the detector on the left-hand side, the subsampling of the image of gold nanoparticles. And again, in this case, it's a Z-contrast image, but you can see a live reconstruction of the image. Now, the advantage of this for doing 4D STEM experiments, whether you want to do tychography, IDPC, or any 4D STEM experiment, is that by getting a live reconstruction while you're on the microscope, and this is the scan as it's progressing, we can change the focus, we can change the area, we can change the location, we can optimize to get the best image, and then we can acquire the full image after that. And that means it's much more practical to be able to do 4D STEM imaging in a microscope when you can see what's actually happening in the system as it takes place. Now, another advantage of using our dictionary is that we can also do image correction. So what we have here are two sets of images. At the top, it's a live image being acquired from the silicon dumbbells again, learning the dictionary and doing a reconstruction. And if you zoom in and you look at on the right, you can see there's a slight misorientation in the sample, there's a slight astigmatism, and we're not resolving the silicon dumbbells in that case. At the bottom, what we've done is take a simulation of the silicon dumbbells that you would expect under the imaging conditions of the microscope. And in this case, we're not subject to the signal to noise, we get a high quality image. And if you now do the reconstruction of your image, you actually get back to getting your silicon dumbbells. So effectively, by using the dictionary while we're working on the microscope, and I'll show in a minute what we can do after we finish on the microscope, you can correct for minor errors in the alignment or the acquisition of your images in the microscope. Another bit that we can do with the transfer of the dictionary is that when we're doing electron microscopy, we can get multiple signals simultaneously. So in this case, what we've got in an SEM image is a backscattered electron image at the top, very high signal to noise, large cross sections, great images that come out. We can get our dictionary, but then that dictionary we can apply to an EDS map. So this, in this case, this is the aluminium edge for the aluminium peak from the EDS map. And using the dictionary from the backscattered electron image, we can reconstruct that map and get something which is a lot higher resolution in terms of what we can see. So effectively, by using the dictionary transfer that was developed by Sensei, we can take these low signals and we can get a much higher quality image for the same amount of time and acquisition process. Now, the other things that we can do with this is that after we've got the images, and you may have noticed in those images that I was showing are coming live, they're a little bit blurry. And the reason they're a little bit blurry is that effectively what we're doing is we're running the reconstruction at the stage where we're trying to get speed. But after we've acquired the image, we can then process or reprocess the images and focus on getting the highest spatial resolution out of it. And because the images are always the same, they're using the same dictionaries, we can actually get a much better reconstruction that comes out. So here is an example of that. We have a 25% reconstruction again, using those those gold nanoparticles that we were looking at. We can separate the dictionary elements out from the background to the particles, and then we can recombine the dictionary elements together again, and this time focusing on taking the whole thing through to convergence, then focusing on the high resolution information that comes out. And you can see you get very, very high resolution information from those gold nanoparticles, and you can even see the individual gold atoms that are sitting in between the two nanoparticles. Now, the implementation of SenseAI is very straightforward. Essentially, we can plug our control software either directly into the scan generator in the microscope, if the scan generator is advanced enough, or we can use an external scan generator and just plug through this external generator into the external port on any microscope, and then off you go. It's basically plug and play, and then a system will allow you to get your subsampled images. So with that, thank you for your attention. Here is the contact information if you want to learn more about SenseAI, and thank you very much.
Okay. Now that we have run through the initial presentation and introduced you to the technology, we are going to show a short demo video here.
Here is a demonstration of Sensei being used to inpaint a subsampled dark field STEM image of a pair of gold nanoparticles using only 25% of the input data. The two core technologies on show here are subsampling and dictionary learning based inpainting. Subsampling is the act of acquiring intentionally incomplete data as seen in the center image, and dictionary learning is a method of learning the spatial components of a piece of data in the form of a dictionary. This can be seen on the far left, where a dictionary is being learned in real time from the subsampled data in the center. As time passes, the algorithm finds the common features of the image and converges to a solution of what a set of structural features are present. The dictionary is then combined with the subsampled data to produce a reconstruction, which can be seen on the right. The dual application of these techniques allows for more efficient data acquisition, meaning less time and dose is used to acquire the image, and a reconstruction of the object is produced for immediate validation. Both of these methods can be employed using SenseAI's user interface, which is easy to use and flexible enough to allow for both simple and complex data acquisition and reconstruction processes. These methods aren't only applicable for the imaging of periodic crystalline materials, however, and shown here is an application of SenseAI to an image of an algal cell produced by cryofibsem, and was imaged using only 12.5% of the pixels of the equivalent full-frame image. Despite containing no periodic structures, the reconstruction is successful. This is because the dictionary learning algorithm doesn't learn periodicity, but spatial redundancy. In this case, the sample is sufficiently spatially redundant to allow for those key spatial features to be learned easily. For applications such as these, the imaging of biological materials, high-dose imaging simply isn't possible, and so pushing the limits of the system using subsampling and painting is incredibly useful in maintaining high-quality data without destroying the sample. As noise is often incoherent to the sample structures, dictionary learning methods can also be used as denoising tools. Shown here is a low-dose, high-resolution TEM image of a polymer crystal, a highly beam-sensitive material which is difficult to image without damaging. In the bottom left is the dictionary learned from that noisy image. On the right is the denoised reconstruction. This denoised result is easier to analyze and can be produced at the point of acquisition using SenseAI. Finally, here is a demonstration of using a webcam as a mock scan generator. First, we can see what imaging using a traditional raster scan might look like, and at these settings the frame rate is rather slow. By only changing the sampling pattern and sampling rate, an immediate frame rate increase can be seen, and at 25% sampling, this corresponds to a 4 times increase in frame rate. As time passes and the algorithm is allowed to run, the dictionary converges towards an optimum solution, increasing the quality of the reconstruction. Even as new objects are brought into the field of view, the algorithm is quickly able to adapt. This increase of frame rate is incredibly important for the imaging of dynamic phenomena, but also just as a method for improving the responsiveness of imaging systems during alignment and just making the imaging process in general feel smoother. Thank you.
Okay, now that you have seen a quick example of what the workflow looks like, we'll share the case study and dive into how this actually affects workflows in labs.
So I'm Professor Roland Fleck. I'm Academic Director of the Centre for Ultrastructural Imaging at King's College London. So King's College London is a Russell Group university located in London, United Kingdom. It's a major teaching and research centre with an international reputation. We cover everything from medical and the life sciences through to chemistry, physical sciences and the arts. And the Centre for Ultrastructural Imaging, of which I'm Academic Director, is its core electron microscopy facility here to help support the broadest range of research and academic adventure as possible. So my personal interest in research is really focused on instrument development and innovation. That's really to allow the facility, Centre for Ultrastructural Imaging, to progress and remain current. The major challenges we've encountered is really relating to charging artifacts, sample stability. Our preferences were to work with vitrified cryo-fixed material, which is essentially frozen. It's extremely beam sensitive. If you put too much dose in, you will devitrify your sample. It doesn't have any introduced contrasting mechanisms. We don't use heavy metals. So as a result, it's a challenging sample to work with. And that's why we've been approaching SenseAI to try and resolve some of our technical challenges. In terms of cryo-electron microscopy, it probably depends upon your starting position or your perspective. In transmission electron microscopy, the cryo approaches are fairly well established, particularly for structural biology. The Nobel Prize was awarded in 2017 to Richard Henderson, Jacques Dubochet, and Joachim Frank. So in that field, it's considered essentially an essential tool and is fairly well adopted across the research community. In the large volume imaging community, cryo is still really in its infancy. The majority of groups would use chemically fixed, resin infiltrated, and heavy metal contrasted samples. And they would be looking to use serial block phase, so essentially a microtome located inside the SEM chamber to section and then image repeatedly over an extended period of time, or fib milling, where essentially you use an ion beam or a plasma beam to sputter away or remove your top surface and then image, or an array tomography approach, where essentially you would do all your sectioning external to the microscope, load a large number of individual sections, and then image them sequentially. But all of these are based upon the room temperature resin infiltration approaches. The challenge with all of these, albeit them extremely stable once you get into the microscope, is they are sensitive to processing artifacts, particularly changes in your lipid membranes can be disrupted by the chemical fixation approaches. They are subjected to quite severe osmotic stresses during the processing, which can change the shape of organelles. You disrupt some of the organelle structure and organization, and soluble components can be displaced, moved, or lost from the cells. Essentially, you are trying to interpret the image based upon what you've done to the cell in preparing it for the electron microscope, which is unlike cryo. In the cryo techniques, we rely on vitrification, so very, very rapid cooling of the cells to essentially move water from its liquid state to a metastable state without the formation of ice crystals. And essentially that preserves the cell in its living native state. All the membranes are essentially preserved without further osmotic stress, so they should be as close to how they were in a living state as humanly possible. So one of the major challenges for VEM, volume electron microscopy, which is really a rapidly developing field, is the size of the volume which people wish to achieve. Essentially, the bigger the volume, the more informative it is. Essentially, because you're wanting to look at all of the cellular components and perhaps even multicellular environments, so you can understand how each individual cellular organelle or structure relates to its neighbor or associating organelle, and how each individual cell, and cells come in all different flavors, sizes, shapes, and types, how they all interact together to form a functioning tissue. So this is really where volume electron microscopy is extremely innovative and valuable to understand these larger interconnecting relationships between cells and organelle. Obviously, the bigger the volume, the larger the data set, which makes processing extremely challenging. And in addition to that, the larger the volume you wish to acquire, the longer it takes. So we have a challenge in terms of total data volume and the time required to acquire the volume, which in itself puts a pressure on the microscope because it has to remain stable, functional, and operational for very long periods of time. And this could be measured in weeks in many cases. So SenseAI has two advantages in this area. One, the compressive sensing approach essentially acquires smaller data volumes, which helps with the post-processing of data. But one of its major advantages is if I'm only collecting, say, 20 or 25% of the field of view by using the compressive sensing strategy, I'm essentially working 4 or 5 times faster than I can otherwise. So the time required to complete an entire volume is significantly reduced, which is less stressful on the instrument, less stressful on the operator, and generates much higher success rates. So one of the other challenges I have, particularly with focused ion beam scanning electron microscopy, the same would apply to plasma sourced instruments, is that the milling material is essentially being used each time we turn on the instrument and we're milling away a surface. That comes with a significant financial cost. If I can acquire the same volume in a quarter or a fifth of the time, my source lasts 4 to 5 times longer. So it's quite a significant difference in terms of the total cost to generate a single volume, which is an added advantage of using the SenseAI approach. I think volume EM has been developing extremely rapidly over the past maybe 5 to 10 years, certainly the last 5 years, and it's become incredibly valuable as a basic tool in life science research, notwithstanding the challenges of sample preparation and artifacts being introduced. If cryo volume imaging can be made robust and routine, and certainly the instrumentation, which I have, is extremely robust, with the aid of SenseAI to help with signal to noise, damaging artifacts, charge balancing, and speeding up the entire process, you could envisage a situation where cryovolume EM becomes routinely practical and simple to apply within a laboratory environment. If that's the case, then I could see wider adoption and more labs employing it. Certainly high pressure freezing to vitrify your sample, or plunge freezing to vitrify cells on grids, is routine and very well established. Cryosample transfer can be conducted cleanly and robustly if your laboratory is sufficiently well equipped. The FIB systems are also extremely stable and well engineered today. Really, the challenge is handling the vitrified sample and the difficulty of actually imaging it and generating sufficient signal to noise to allow you to reconstruct your volume and analyze it in a meaningful way. And SenseAI is essentially opening that door to us at the present moment in time. Well, in principle, the SenseAI approach, where I can simply use it to speed up the process and manage the imaging part of the data volume generated at the end of a volume acquisition, makes it applicable to any scanning modality, which would apply equally as well to room temperature FIB-SEM, serial block phase, or array tomography if you're using the SEM to collect the data. It also has large numbers of opportunities in STEM, which can also be employed in a cryogenic environment. So I think as a broader tool, if it's easily accessible to the end user and perhaps somebody who's not a computational expert, it opens up the avenues for the compressive sensing and SenseAI to be essentially routinely employed as an acquisition tool on any microscope that's suitably equipped.
Okay. We're going to open our Q&A in just a moment here, but before that, I have one question for the audience, and this is just to help us gauge how best to follow up with you after this webinar. So if you are interested in acquiring the SenseAI technology for your lab, feel free to select any of those options. We obviously have experts who can talk a little bit more about the technology, maybe zero in on exactly how this might benefit your research group the most. If you'd like to speak to one of those application experts, feel free to select that option. If you're just here exploring technologies for your lab, thank you so much for joining us. We're glad that you were here. We hope you learned something. And if you have questions, Dr. Browning will answer them in just a moment. And I'll leave our poll question up for another few seconds, and then we'll start answering our questions. And in case you're wondering where to enter your questions, there is a questions tab at the bottom right-hand corner of your screen. So you can click there, type in any question that you have, and we'll read them out live here in just a moment. Okay, at this point, I'd like to welcome Dr. Browning to the stage, and we will begin our Q&A. Okay, hello, Nigel.
Hi.
Okay, so let's start with this question. This is a very interesting one. Does the dictionary grow continuously during imaging, or is it always 6 by 6 elements, as was shown in the demo on the left-hand panel of the screen? That is, does the quality of reconstruction improve during the process?
Okay, well, so the approach is actually to try and minimize the number of dictionary elements, because the more dictionary elements you have, the longer it takes to get the reconstruction. The number that we have on the screen that you saw in all of the examples is what we typically find is best for reconstructions that we've done so far. Now, that's different from the quality of the reconstruction improving with time. So it's not that it refines the number of dictionary elements, but the accuracy of the dictionary elements is refined. And so if you notice in the reconstructions we were showing, that panel of dictionary elements would actually have the dictionary elements changing. And so with every iteration, they become more and more burned into the right structure needed to do the reconstruction. And then the reconstruction becomes quicker and quicker because of that.
Thank you. So this is referring to something that you said towards the end of your presentation. Does SenseAI come with its own scan generator, or can it work with any existing scan generator?
Yeah, so the Sensei is really a software company and develops software to go through it. We don't make any of the hardware ourselves. That being said, what that means is that we can integrate into any system that is there. And whether it's an external scan generator or whether it's an internal one, it all depends on the stability and the speed of the scan generator. So we tend to move with an external one if the inbuilt one within the microscope isn't sufficient to get to the fastest speeds in terms of the resolution. But there is no limit to the hardware that this can be applied to. It's completely agnostic to which bit of hardware is being used to form the scan.
Okay, hopefully that answered this person's question. Let's see. If you have any pressing questions, feel free to drop them in the question box. I know there was a lot of information presented here, so some people might still be digesting a little bit everything that they've seen so far, but since we have Dr. Browning here, we're happy to answer any questions that you might still have. This is a good one. How will the reconstruction handle anomalies such as defects if these defects are not within scanned areas of the sample?
Okay. So the way that a dictionary works is that it doesn't form a template of the image. So there's a lot of image processing techniques where you are essentially forming a template and you match that template. And so the problem then is you have an issue if there's something different in the structure. You also have a problem with defects if the defect itself is something that is changing or not in your training set, if you're going to use a post-acquisition form of machine learning or artificial intelligence. And this is the approach where you have a lot of training data. If you don't have the right training data, then the software can hallucinate. None of those things apply with a dictionary. And you know, the idea is that there are different works of fiction in English that all use the same dictionary. The dictionary is the same, but the work of fiction or the song or whatever it is completely different. And that's exactly what happens in these images as well. What the dictionary does is understand the dictionary elements. So if you take the example of the silicon image, the dictionary elements for a silicon defect will be the same as the dictionary elements for a silicon perfect structure. They will just be used in different weights and in different positions to reconstruct the full image. So invariably, there is not a problem in doing defects. Now, if the defect is at a slightly higher resolution than the image that we've got, so let's say the spacing is smaller than the resolution of the microscope, what happens in dictionary learning when you have something where you don't have the resolution in the image to see the thing that you're looking at, then what it does is it just gives you a blurry space. And so the way that we approach something like that in terms of the compressed sensing and the dictionary learning is that would tell you that's the area where you need to sample further to be able to get more information to reconstruct the image. And that's a form of compressive sensing or sparse sampling and in-painting that's called adaptive sampling. And with the sensing approach, we obviously try and do it with a single image because you go much faster with that. But you can also do adaptive scanning. If the first image doesn't give you the quality of the image to reproduce everything in there, you can go ahead and completely adapt the scan to increase it. Now, of course, if the resolution is below the resolution of the microscope, you're not going to be able to reconstruct that. But it doesn't create something there to fill in the space. It just gives you a blurry reconstruction that doesn't show any feature at all.
Very good. Okay. A somewhat related question, also talking about the dictionaries. When acquiring multiple similar images, can dictionaries be reused, for example, as starting values to speed up the subsequent images?
Yeah, so one of the technologies that we implemented in SenseAI was basically dictionary transfer. And so that was one of the examples I showed of transferring a dictionary from the backscattered electron image into the EDS map. And so you had much higher signal to noise from the backscattered image. And so you could use that dictionary to reconstruct the EDS map. And that works in scanned images because, of course, as a function of the beam position, it's always going to get a registration of whatever scattering, whether it's EDS, EVSD, whether it's a 4D stem, whether it's EELS, all of those things are as a function of the same probe position. So effectively, you can transfer the dictionary across. The other one that I showed was the transfer of a dictionary from a simulation into an experimental image. So a simulation has essentially no noise on it, so you get very high signal. And so you can transfer the image across from a simulation with high signal to noise onto an experimental image where you have more noise associated with that. And that allows you to fix some of the artifacts that come into the imaging system. Again, it's a dictionary method and not a template matching effort. And so effectively, you're not forcing the image to fit to your dictionary is interpreting your image. So absolutely, we can transfer dictionaries across. And, you know, if you had a microscope or you had a lab where you were working on the same sample continually, I mean, a lot of us look at the same samples, then you can build up that dictionary that will work for those samples on your instrument. And so all of that will be taken into account into forming a dictionary which is learned from multiple images and multiple applications. And all of that is part of the SenseAI approach.
Okay, thank you, Nigel. Can SenseAI be applied on cryo-electron tomography pipelines?
Yeah, so in the sort of running through the software, Danny showed an application to a TEM image where you can denoise. So effectively, it can apply to the TEM side of it in a form of denoising. So you can use that through the pipeline. The one that we are working on more at the moment is the application of 4D stem to that cryo-EM pipeline. So solving with 4D stem or solving the images with 4D stem and applying that through a standard pipeline. But essentially, it does help on all sides of it in terms of reducing the number of images because you get higher signal to noise.
Okay, is there an API or MCP available for the software? I'm considering the aspect of integration within, for example, serial EM for STEM work. Oh, that's it. Great question.
Yeah, so we obviously have to, for Sensei to work, integrate with all of the APIs from all of the manufacturers to make that work. There is absolutely no problem with providing that for Sensei as well, or from Sensei. I mean, one of the things that we have as part of the coding part of Sensei is that if you wanted to introduce a different algorithm into the system to run instead of using BPFA, we can also handle that as well. So obviously, it's a case-by-case basis, depending on what the software is being used for and what the overall plan is. But there is, in principle, absolutely no problem in doing that.
Great. Let's see. Could any image drift or vibration, for example, in a cryo-TEM image, affect the image reconstruction?
Yeah, so I mean, obviously we can't fix the problems that you have in electron microscopy. We can't make a blurry image that's out of focus into a magically focused image, and we can't overcome the problems with drift either. We still need to do drift correction. The point of it for the Sensei side of it is because in a STEM image, we are increasing the speed of the scan by a factor of 10 in terms of going through that system, then any drift will be 10 times less important if you're going to your image to be much faster. So effectively, we do reduce the effects of drift because we don't have to worry about acquiring for so long. Each individual image is acquired much quicker. And so that means that it has less effect from the drift. And then if you want to then use a drift correction on that, you can then use that with obviously much faster speed of the individual images to overcome that problem.
Right. How are the random or pseudo-random qualities of the scan pattern achieved? That's a great question as well.
So essentially, what it is a software will create a map of the scan, which is then basically passed through the scan generator into controlling the microscope. And so this is a standard way that everyone does this to create a scan, except the difference is the Sensei software creates different patterns rather than just the regular side-to-side stem scan. So we can create all kinds of different patterns that go through that. There is an issue that we have to do for the fastest scans, which we have to overcome the issues of hysteresis in everyone's scan system. And so what this means is that you can't do a random scan where you go from the top left corner to the bottom right, back up here and over there, because the time for it to settle would then be so long that it would drift further and you'd have problems with that. So the way that we create a pseudo-random scan in the microscope is to do a random walk in terms of that. So each randomness is only plus or minus 1 pixel in any direction, and we create these lanes. So if you had a lane where you wanted 10%, you'd have plus or minus 1 with bounds of plus or minus 10, and so this thing would oscillate and create a pseudo-random walk type application. And so that will apply to all of the situations when you're going for really high speed and you're worried about hysteresis in the scan. For techniques where you're going to have a longer dwell time, so EDS, EELS, 4D STEM, there you can just create a random scan of just moving random positions in a scan, going to the next line and random positions there. And so there it's actually very straightforward to create the random scan.
Very good. We were just talking about EELS. The next question is related to EELS. They're wondering, did you try it on EELS spectrum images? In that case, would the dictionary contain both spatial as well as the EELS features?
Yeah, so we haven't done EELS just yet because it's the 4D STEM is the next implementation, which is coming in the next few weeks. After that is to go on to EELS. So in the EELS approach, you can actually use the camera as a subsampled scan on the spectrum itself, and you can use regular compressive sensing techniques on that. And that means each individual spectrum can be acquired much quickly. You then have the position in the STEM probe, which you can reduce as well. So effectively, you would have two sets of dictionaries in that case. One is when you converted it into a map, which is your 4D, which is your map of the of whatever it is, the edge, the oxygen edge, whatever it is for creating a map. But you also can use it to create a dictionary for your one-dimensional spectrum. So there you can actually use compressive sensing to reduce the noise and increase the signal to noise on any individual spectrum. So at each location of the acquisition, we would acquire multiple spectra, maybe 100 spectra at each position. You can then do very much the same way as CMOS detectors we use in structural biology. Because you've got so many individual spectra, you can look at the place where damage comes in. You can see where that is, sort out the ones that you're going to use, bundle them all together and get a dictionary for the full 1D spectrum, and then use that to interpret all of the individual spectra. So there are very straightforward ways of using dictionary learning for EELS maps and also for EELS spectroscopy. And that's the thing that we will start developing probably in the next 2 or 3 months.
Okay, so to this person, stay tuned. Subscribe to the Sensei newsletter. That way you'll be the first to know when the EELS program comes out. Okay, how many BPFA iterations are computed when scanning live? Has the algorithm converged? And after the session on the microscope, can you rerun the BPFA algorithm with more iterations?
Yeah, so we typically use a single iteration going through when we're running live. Sometimes if you're looking at the same type of material, you're looking at the same, let's say the same crystal in the same orientation, your dictionary will continue from one image to the next image and going on to that. So it will effectively iterate going through it. But this is the point I was saying in the presentation, where if you look at our fast live images, they look a little bit blurry. And the reason for that is that effectively we're using the lower spatial frequency dictionary elements to fill in the intensity very quickly, and that gives us a slightly lower resolution. Now, the way to think about this is very similar to something like tomography, where when you do a back projection in tomography, you oversample the low spatial frequencies and undersample the high spatial frequencies, and then you do something like a weighted back projection to fix that problem. With the dictionary learning, we get enough information out to be able to do a live focusing, a live analysis of the image, so that when you're on the microscope, you can use that. But then after you finish on the microscope, you can go back and completely reprocess all of the images, because essentially you've got enough information to see that. That's what we acquire. And so that means you can go ahead and check, you can run through as many iterations you want, you can use different algorithms, you can use different parameters to do the fitting, you can optimize those. We have a set of parameters that we recommend people to use, but obviously there's a plus or minus in that for any particular application, you might want to check that. And all of that can be done offline. Effectively, what the subsampling means is that we don't lose any information. All it means is that we are acquiring the minimum information in any experimental acquisition that allows you to get a full image at the end. So it's a minimum amount of data, but you still have a full image. So once you've got the full image, you can go back and rerun the dictionary learning. You can run standard image processing on those. You can put it all through an artificial intelligence or machine learning approach if you want to denoise that way. All of that, once we've created the fully sampled image, is completely applicable to the images that are there, because at that point, they're 100% sampled images.
Okay. Let's see, our next question is the beam blanked between spatial positions in order to reduce the dose?
So in this case, because we do a continuous scan, we don't do blanking. So there's no need to, because it's scanning continually and it's stepping. In the same way, in a normal SEM, you don't typically blank in between the positions in the scan. Now, blanking in itself has a cost associated with it, which is a temporal cost. But if you blank as you move, the beam has to, when you unblank, the beam has to come back to that position, and then you have to acquire the pixel that you want at that point. So for the fastest temporal resolution, we don't blank, because it's just a continual scan with a random walk that goes through that. But potentially, if you had something you very precisely wanted to control the dose, there is no reason why you can't use the Sensei software along with a beam blanker. All it means is you will have an effect on the speed of what you're going to get out in the final end because of the cost of doing the switching of the beam off and back onto the sample. So all of that is, again, applicable through the Sensei software. We don't use it for the fast scans at the moment, but there's absolutely no reason why it couldn't be incorporated into any workflow for STEM.
And to the person who asked this question, if you'd like to discuss this more, feel free to reach out and we can answer your particular application related question. Maybe it's related to their material, whatever the case may be, we can find a solution that works for you. Okay, it looks like we're coming to the end of our questions here. So if you still have a question, please do type it into the box so that we can answer it here. But in the meantime, I will once again share our contact information while we wait for the last few questions. Like I said, you can always explore our website to read more about this technology. You can call us at our headquarters or send us an email with your specific question related to your material, or perhaps it's the instrument that you have. We're happy to answer any of those questions. I think we'll give just a little bit longer for anyone to drop their last minute questions in, but I'd like to take this opportunity to thank everyone for joining us. I'd like to thank you, Nigel, for joining us as well. This has been a really great and engaging session. I personally have learned quite a lot about the Sensei solution, so I'm hoping that our attendees feel the same.
Yeah, well, thank you. And then I'll give my thanks to everyone for attending as well.
All right. Well, thank you, everyone. I hope that you have a wonderful rest of your day or evening, depending on where you are in the world. And again, thank you, Nigel. Thank you, everyone. And we'll be signing off here.
Thank you.
Thank you. Take care, everyone.